Get away!
Calm down, Noi. Don't worry. It's not easy to get virus because our body has immune response against virus.
Immune response?
Yes, let me tell you about it.
The virus is an organism that can replicate only inside the host cells. Virus is independent particle, consists of two parts. First, the genetic bacteria made of either DNA or RNA. And the second, protein code that surround and protect the genetic material. In some cases, they are an envelope of lipids that coating when they are outside the cell. Virus can infect many organisms such as animals, plants, and bacteria. The steps of viral infection are attachment, penetration, viral replication, and immunopathology or cell damage. In humans, virus can infect into our cell by firstly, virus invade host cell through first-class barrier such as epithelium cell and mucus. Virus attack host cell by using ligand as the key, fit to the receptor on the host cell as a lock. After that, virus can replicate itself and produce viral protein and viral nucleic acid in host cell. Viral nucleic acid is captured by toll-like receptor 3, 7, 8 on endosome. Then, infected cells secrete type 1 interferon alpha and beta to induce an infected cell to become antiviral state cell.
Oh yeah!
These antiviral state cells allow virus entry but block viral release by inhibiting viral synthesis, degrading viral RNA, and inhibiting viral gene expression and viral assembly. Therefore, viral particles can be destroyed by mechanism of type 1 interferon alpha and beta. Don't forget, infected cells still survive here. Normally, NK cells work alone like security. NK cells need balancing signals of ligand activating receptor and MHC class 1 inhibitory receptor from normal cells. However, virus inhibit expression of MHC class 1 on the infected cells. After that, NK cells release perforin for permeabilizing cell membrane and gamma for degrading protein inside the infected cell. Finally, infected cells are dead. Moreover, NK cells secrete type 2 interferon gamma to activate macrophages to become phagocytosis killing cells. During viral replication, virus can also destroy infected cells. Free viral particles might get outside. Then, antigen-presenting cells of APC recognize free virus and capture them. Finally, antigen-presenting cells will present viral antigen on MHC class 1 molecules. Interferon gamma from NK cells enhance antigen-presenting cell efficiency by increasing MHC class 1 expression. Next, antigen-presenting cells might get to lymph node and present viral antigen to naive T cells. In the lymph node, the interaction of naive T cells and antigen-presenting cells occur by T cell receptor and MHC peptide complex. Co-stimulatory molecule interaction B7 and CD28, and cytokine from antigen-presenting cells interleukin 12. Then, naive T cells release interleukin 2 for stimulating proliferation themselves. To to become cytotoxic T cells. Naive T cells are differentiated by two signals: T cell receptor and MSC peptide complex, and cytokine from helper T cells. Cytotoxic T cells migrate from lymph node and go to infected site. They can eliminate infected cells by perforating gamma pathway and apoptosis pathway by fast ligand. Macrophage phagocytosis viral antigen and present viral antigen on MSC class II molecules. Then, macrophages go to the lymph node for presenting viral antigen to naive T cell by T signals. T cell receptor and MSC peptide complex, co-stimulatory molecule interaction B7 and CD28, and cytokine from macrophage interleukin 12. To activate naive T cells, produce cytokine interleukin 2 for stimulating proliferation and differentiation to be T helper 1 cells. That secrete type 2 interferon gamma for inducing macrophage to be activated macrophage and stimulate activated B cell for isotype switching. For the specific antigen production, free antigen or antigen presenting cells activate naive B cells to proliferation and differentiation to be short-lived plasma cells. That secrete IgM. When type 1 helper T cells stimulate activated B cells by CD40 and CD40 ligand interaction for isotype switching. B cells will go inside the germinal center for affinity maturation by follicular dendritic cell and follicular helper T cells. The activated B cells become memory cells and long-lived plasma cells with isotype switching and high affinity maturation. Here we show how antibody eliminate free viral particles and infected cells. First, neutralization, which prevents virus entering host cells. Second, opsonization, capture viral particle and induce phagocytosis. The other is antibody-dependent cell-mediated cytotoxicity. Bind to viral antigen on surface of infected cells and induce NK cells to eliminate. When our body gets viral infection, our immune system tries to eliminate that pathogen. However, virus can express some protein that interrupt our immune system mechanism, so the virus can survive by avoiding our immune response. This condition is immune evasion. There are various mechanisms that virus use to escape from host immune response and cause the disease. Antigenic death and shift leads the virus to be resistant to immune response, such as the avian flu or the swine flu virus. Virus can inhibit the stability of MSC class I molecules associated to presentation of cytosolic antigen. Because of that, MSC class I molecules cannot present peptide antigen to the naive CD8. Moreover, mutation of cytotoxic T cell epitopes also lead to loss of MSC SC class 1 binding and loss of cytotoxic recognition. Some viruses produce molecules such as virokine and viroreceptor to invade the cells without interference. After that, interleukin 10 will be secreted out to inhibit macrophage activation. Virus may infect or either kill or inactive immunocompetent cells. For example, HIV that inhibit the T cell activation by eliminate CD4 T cells.
Oh, I got it. Get well soon, girl.
Okay, thank you. Bye. Thanks for watching.